Welcome to video 2 in this course G05 on antibody analysis. So here we're going to talk about antibody structure briefly and class switching just for anyone who's new to that area. So we talked previously about how hybrid mutation increases antibody diversity, widening the diversity of antigens to which these different antibodies will bind. We also have to consider things like affinity maturation, which means that B cells that produce antibodies that have lower affinity are therefore less useful, tend to undergo apoptosis, whereas those with higher affinity will survive and produce more antibodies that's more useful to the organism and the health of the host. So when we consider the effects of hypermutation, we can actually find that there's really diverse antibodies that are made. And although they have this kind of shared Y-shaped structure with the two heavy and light chains there with the constant regions, heavy and light there too, we can find that there's great diversity there. So this structure shows IgG, and it has the heavy chains shown there as part of the FV region, and we have the constant regions, and this variable fragment is the molecules to which it binds are determined by the heavy chain light chain interactions. So we have immunoglobulins M, D, G, A, or E for the H chains, and IgL and K for the light chains, lambda and kappa. The three CDRs compare to determining regions in the heavy and light chains, and these are the regions that bind G antigenic epitopes, but they vary in size. So they're about 14 amino acids in humans, whereas they're about 26 in cattle and 11 in mice. And we know that the CDR3 region is the most variable of these. And the example in terms of viral responses to exposure to H1N1 flu vaccine. This is determined majorly by IgH specificity, and that's mediated by symbiotic mutation and the efficacy of this clonal expansion affinity maturation progress. So that kind of are two key factors that underlie the variability that we see in the host response. If you look further at the detailed molecular structure of this, the FV variable region, we can see that the CDR regions there are highlighted here. So we have CDR 1, 2, and 3 highlighted. And we've got 3 loops of the variable light and heavy chains. And we can also talk as well about class switching in terms of the VDJ recombination process as well next. So we have this process of VDJ recombination. And we see here the V, D, and J alleles as well as the constant regions at the 3' end. Then we have our selected VGJ allele, and then we have our constant region here as well. And this example here is given for IgM. So this is the IgH locus that encodes the Ig heavy chain, and this determines which antibody type will be made. So on the cell surface, you get IgM and D expressed with initially identical antigen binding regions, but their naive cells as they mature, the daughter cells will be different. So in this diagram, what we're looking at is class switch region, CSR. The characteristics, they're repetitive regions of about 1 to 10 kb. It's G rich. And then certain elements, S stands for the switch regions, mu, alpha, and eta have 5B repeats, and the gamma chain is 49 base pair repeats. So we know that this constant region is changes in the variable region doesn't at this point, and the CSRs are usually at the 5' end of the class switch region. So what we'll end up with at the end is this, the class switch product here is the L at the end, and then the deleted region is left over as well. So that happens after the DNA at the 2 S regions here are broken. And this whole segment is taken out. Sorry, it's extended, is taken out. The DC, this link is repaired by non-homologous end joining. And then we have for the constant region, we've got 5 different isotypes: ADG, ADE, G, and M in humans, and then in other organisms might be different. If we look at a more complex example, you can go to this link for this example. We can see here we got the human IGH locus, and we got the BDJ allele selected here, and then we have the S region, another S region, another S region, another S region, and so on, another S region, and these constant regions here as we go through. We have aid and activation-induced cytosine deaminase, assisting with this process of forming this loop so that 2 S regions can come together to excise the constant regions that are not of interest and create this segment here that we see with the BDJ element and also the constant region of interest. And then we have this excised part as well after this. The 2 breaks that are created are resolved there. And this process means that we got different types of antibodies that are produced, and they have different behaviors in short. And this use of summary diagrams like this, which outlines the secreting nature of IgA, for example, the numbers of antigen binding sites, which again differ across all of them. Their molecular weights all differ. And in human body sera, you see that it's mainly IgG and then some A and M, but less so for D and E, unless there's a particular something happening inside the body. So that's a brief outline in terms of antibody structure and class switching. Of course, there's more there's more resources that expand this, and this is just a brief overview.